be here. Thank you. Okay, this is the right thing. Okay, it's a pleasure to be here. It's my first time in this college, so I hope to be back in the future again. Today I'm going to talk about something completely different to what you have seen so far, I'm sure. And I'm going to talk about the fruit fly, Drosophila melanogaster, and how we use this little insect to understand many fundamental problems in biomedical research. But before telling you some examples about what can we do with this fly, I will give you a brief overview of the biology of this amazing insect. Why Drosophila? Well, there are many advantages that the fly provides us in the lab. For instance, it has a very small genome. It has about 13,000 genes compared to the high complexity of other genomes, for instance, in humans. That is an advantage for us. It has a short life cycle. Usually in 10 days, you get the following progeny and so on. So we don't have to wait a long time, like if you were working like with mouse models, for instance, you have to wait longer. So we can have a large number of progeny per mating pair. And this is important because, for instance, a single female can lay up to 100 eggs in one day. Okay? So imagine that you can have several females in a vial, so you can get literally hundreds of progeny to investigate or to study the question you have in mind. Okay, that's an advantage. We can generate transgenic flies easily, very easily. And this is, you can have a gene, let's say from a human or from a mouse or from any source, and you can put it into the fly by following a specific protocol to inject that DNA in fly embryos. And then you generate transgenic flies by that procedure. It's pretty standard procedure, and we do it in the lab all the time. And importantly, many aspects of human biology are present in flies. Okay, so sometimes when I'm talking with people, in restaurants or somewhere else, what do you do? I work with flies, and so what? With flies? But I say, what do you do? Well, I analyze the brain. Do they have brain? Really? And most people think that they don't have these things, but they have brain, heart, and gut, and respiratory system, and many of the behavioral traits that you see in humans, like aggression or drug addiction, alcohol addiction, or sleep disorders. They snore actually. Believe it or not, they snore. They have. You can study all these things in flies. It's amazing. So these are some of the reasons why we work with flies. So this is just to give you an idea of how the life cycle occurs. We have female and male. Male is on the right. The male is a little bit smaller than the female. They mate. The females deposit an embryo, usually on soft surfaces, that's why they like to go to fruit, and they deposit embryos right there. 24 hours after the embryo has been deposited, then they go through 3 stages where they become, the larvae hatches, and then you see like a little worm-like structure. That's the larvae. It stays for 3 days. For 3 stages, known as first, second, and third instant larvae, and then they became pupae. Okay, and stay for another 2 or 3 days, and then finally the flies close from the pupal case, and they start the cycle all over again. So once they close, 8 hours or so, they are ready to mate again. Okay, so they are sexually mature in more or less 8-10 hours for sure. And this is also an advantage because when you're working with mouse models, okay, you have to wait sometimes 6 weeks till they reach, they are sexually mature. So this for us is we can just get flies and start experiments even the same day. It may do crosses and all that. So that's pretty nice for us. Another important thing is that the fly has been in many biological laboratories for 100 years. Okay? There is a lot of information about the biology of the flies, about the anatomy. We know with extraordinary precision many details about the fly. We know what you can see here. This is the digestive system, the muscles. We know all the muscles, where they are, where do they connect. This is the respiratory system. We know the trachea, the nervous system. We know the brain is here in gray. And then the peripheral neurons are all in blue. So we know a lot of information. Okay? There are books and drawings and a lot of information regarding the anatomy and the biology of the flies. We can also perform highly advanced imaging studies to visualize the body, the inside of the fly. And just for comparison, I mean, we have very advanced procedures of MRI scanning to visualize the morphology or anatomy in humans, which is shown right here. Similar to that, we have analogous procedures to see what's going on inside the flies. And this is a movie where you are going from top to bottom, and you see there's the muscles right here. You know, see the many other structures here. This is the brain, and so on. So this is something that is pretty nice, because whenever we are studying a problem or a disease, we can just do this kind of imaging and see what's going on inside the flies. So what do we need to work with flies? Okay, this is what we have in my lab. You need basically a fly room where you have a stereo microscope with lower magnification. That's what we use on daily basis. Okay, so we have some kind of sleepers right here where you have some gas. So you pour the flies right there, then you have a pedal with the gas is coming, so they smell, and then they immediately are anesthetized. So they fall down to sleep, so you can manipulate, otherwise they will fly away. Okay, so that's what we have. Then you need a kitchen. We have a kettle where we just mix all the food, all the components, and then there is a peristaltic pump where you pour the food in these vials or bottles. Then we have incubators with specific temperature, usually 18 degrees or 25 degrees or 29 degrees, depending on the experiment that you are working with. And this is the microinjection station. As I said, this is something that we use to generate transgenic flies. And basically what you will see right there will be embryos, which is this is a picture right here, and we should have a needle where we inject a very tiny amount of DNA, of the DNA of your choice, carrying a gene of your choice. It can be a human, as I say, or from animals or plants or whatever you want. And then you can just inject it posteriorly in the embryo, an embryo that is about 30 minutes old, recently laid by the females. And then you let it go, and it will mature and it close, and then you will have transgenic flies coming from that procedure. So again, the Drosophila genome is not too complicated, or the complexity is a lot lower compared to higher organisms. It was fully sequenced in 2000, in the year 2000. Means that we all know now the entire content of genes. The genome has been annotated. We know the name of the genes, what they do, what proteins are encoded by every of these genes, and so on. So it has 4 chromosomes. As I told you about 13,000 genes. And there are several web pages related to Drosophila biology, but the one that is the most important for the Drosophila community is the one known as Flybase. In Flybase there we can find drawing, images, information about the anatomy, about the genes, about proteins, stocks available, mutations available for genes, what are the homologies between humans and fly genes, and so on. So this is an example of a web page known as Homophila. And basically what this web page shows is that almost 75% of the genes that are associated with human diseases are also present in the fly, 75%, okay, which is highly remarkable. And these include diseases associated with cancer, with neurological disorders, with heart diseases, metabolic and malformation, and so on. There's a lot of diseases that can be studied in the flies, because the flies have the similar genes associated with those diseases, okay, which is quite outstanding for many of us. Okay? So despite the differences in the anatomy between flies and humans, you know, they have many similarities. You know, the way some body parts are developed, you know, it's similar. The molecular blocks that regulate or orchestrate the growth of a limb, for instance, is similar in flies and in humans, and this is again due to the functional conservation during evolution, conservation of the genes. So the same molecules that regulate learning and behavior in humans are also present in flies. Genes controlling the cancer can be studied in flies. The response to drugs can be also analyzed in flies. Mechanisms associated with neuronal neurodegeneration it's pretty similar. This is what I'm focusing in my lab. So I just want to show you some examples of some of the phenomena that can be analyzed in flies. So many questions regarding the development of body parts were investigated, I don't know, like more than 20 years ago, okay? And people started to make mutations in genes in the fly to understand whether there are genes that were responsible to activate or direct organogenesis or specific formation of a given tissue and so on. In this case, someone just activated a gene that was thought to be regulating the formation of legs. Okay, they say this gene might be a master gene, is the one that regulates the formation of leg. Let's see if we activate that gene somewhere else, right? So by activating that gene in the antenna of the fly, now the antenna disappears, and instead of that, you get a leg, the fly legs, okay? So in a similar scenarios, other gene was manipulated that was thought to be responsible for the formation of the thorax. Okay, so they made a duplication of that. Let's put another one, and then what you have is now a fly that is known as bithorax. Okay, so there are many examples like this, but another one was the identification of a gene that regulates the formation of the eye. Okay, so the same similar example. Okay, let's activate that gene where the antenna is formed, and let's see what happened. So now if the organism say, okay, I have a gene that triggers the formation of the eye, but I'm just going to have it in the antenna. So then the organism let's say, okay, I have priority to make an eye. I'm going to make an eye right here, and then the antenna, the information for the antenna is gone. So now you have eyes instead of antennas. You have the normal eye, but then you have ectopic eyes, and there were examples where the antennas were, the eyes were coming out from legs or from wings and so on. And this is more related to the field of developmental biology, where we study development, we understand how we have 5 fingers, or why we have dorsal and ventral parts in the hand, and so on. So all these are questions that can be investigated in animal models. Okay. So a different example of the use of the flies is, you can expose the flies to different type of drugs. It can be alcohol, it can be, in this case, cocaine. So there are laboratories that have special settings to work with cocaine, and they get the vapor and they expose the flies to cocaine, and then they investigate what is the effect of giving different amounts of the drug, and they use video cameras in special chambers to monitor what happened. This is, for instance, a video of how the normal movement of the flies in a closed chamber. Usually they tend to go to the perimeter of the chamber, and then when they have 100 micrograms of cocaine, they start to have these weird movements, and then you have the double doses. They are absolutely crazy. Okay, so they are completely they have weird behavior, and so what people working with this, now you have models to see whether you can make mutations in the fly, and so you can block that addiction to the drugs, and this is a very important paradigm for people working on addiction. So we can also study obesity in flies. So the same way you expose the flies to drugs, you can expose them to different types of diets. You can with the high sugar and so on. This is a normal fly, and this is an obese fly. And the way you do that, you mix your specific food with some dye. In this case, is blue, and that's how you know that they were eating the food that you are providing. So you can combine, you can study the effects of insulin or sugars and all these things. So we have ways to work in that and understand the biology of eating disorders. So another thing is concussion is something that is now bringing the attention of the general public. Okay, it's a real problem now. Is particularly in sports medicine. This actually is the movie just came out recently. It's a pretty good movie, and it talks about the story of how all this happened. So in the recent Congress of the Neurology Association, it was indicated that about 40% of ex-NFL players have brain injuries. Okay, and this is a consequence of constant injury, trauma hitting the head. And this is the case for NFL or boxing or other high-risk injuries. But it also happened in soldiers, in the army, or in accidents. But particularly in sports medicine, is a repetitive, impact, and that over and over and over and over. Later on, when they are probably in the 40s and the 50s, the dementia and other things develop because there are some damages in the brain. There are some molecules in the brain that little by little start to give troubles until the situation is pretty serious. So one of the things that we are doing in my lab is we're trying to working on a model for this. Somebody already published this device where you can, it's basically a surface, a wood surface, and you have a spring right here, and you have a vial, and then you can put your flies right there in that little vial. So you just lift the spring, you let it go, and then there is an impact right here. So the flies have this impact. So you can have it done in several times, a few times a day, or every other day, or every 3 days, and you can regulate the strength of the impact. And then you just investigate the brain of the flies and to see what's going on. There are other devices more sophisticated like this, where you use, I don't know if you can see this, a little fly right there. It's a slow motion, but just for you to see how it moves, and it's going to have the first impact right there, and it's another impact over here, and so on. And so it's again, this is more advanced. You can regulate the speed, how many times, only one impact, or two, or three, and so on. So this is more or less trying to, to replicate the injury. And from there, once I say you can isolate the brain, dissect the brain, and staining, and look for the pathology that is also seen in humans. That's the bottom line. So we use this as a small model, because we cannot do that with humans, and to do this with mice is also possible. But in our case, the fact that we can do this with hundreds of flies, it provides you a very good way to do some statistics and understand what's going on without issues with bioethics issues, because the flies, you can work with flies and you can kill them and you analyze and do whatever. You don't have any issue with the bioethics committee. We don't need to get any permission from the institution to work to do these kind of things. Okay? And now I'm going to get more into what I do in the lab. We have models of several neurological disorders. Okay, so these are some of the ones that we have: the myotonic dystrophy, Huntington disease, prion disorders, Alzheimer's, and Parkinson. One important thing is that none of these diseases have a cure. There is no cure for these diseases. They are lethal disorders, and there is nothing that we can do at this point to stop or delay or prevent the disease. So that's a very, very important problem, and we are all, many of us are working pretty hard to address that situation. Today I'm going to just talk about Alzheimer and Parkinson because of time limitation, but this is basically what we do. Alzheimer's disease, well, this is, but I'm sure all of you are familiar with it, or you have someone in the family or a neighbor or someone who at least have issues with the disorder. It's the third more expensive disease in the United States. It costs about $100 billion per year to the government, and there are 5 million Americans currently have the disorder, 12 million all over the world, and it's estimated about 500,000 are affected here in Florida. Okay, 500,000 here in the state. So it's a serious issue here. As I said, there is no prevention. There is no way to cure the disease. There are several labs that are making progress on the diagnosis, but still, we still a long way to go. How do we make the fly model of Alzheimer's disease? Well, the brain of affected individuals, they have aggregation of a protein known as amyloid beta. It's a small peptide, 42 amino acids, and it forms these aggregates, in the brain that are known as amyloid plaques. So they are composed of these aggregates of this little protein known as amyloid beta. So the idea behind making the model is, okay, what happens if we take this amyloid beta from humans and we just put it into the flies. And interestingly, the flies, they don't have amyloid beta. So the flies don't have this protein. I mean, remember that only 75% are conserved. Well, this is one of those that are not present in the flies. So that was good for us because, okay, well, the fly doesn't have A beta. Now let's bring the human A beta from sick people, and then we just trigger the production of amyloid beta in the brain. And to our surprise, we got a model of Alzheimer's. So you can see right here, this is a section through the brain of a healthy individual. This is the Alzheimer patient. As you can see, there are significant destruction, or I would say the degeneration in some areas. You can see here, right here, this gap is pretty wide. And areas associated with memory are gone, you know, in patients compared with healthy individuals. So that's why there are just issues to remember, or to, you know, the learning and memories are affected in individuals. So when we had, when we were investigating what happened in the flies, then we had the healthy flies and the Alzheimer flies, and we see this massive destruction right here due to the accumulation of the amyloid beta, which was not seen in normal flies. Okay? So we had this model, and we have a ways to assay memory in flies. So I don't have time to go to that, but we can investigate whether they have issues with learning, with memory, and we have protocols, procedures to do that, and they indeed have issues with learning and memory. So let me just now talk about Parkinson. Is another devastating disease, and you know, it's trembling associated with involuntary trembling and movement disorders. It's the most common movement disorder. 1.5 million Americans have the disease, and 50,000 new cases every year. Okay, so this is another problem for the U.S. Economy. So there are several genes associated with Parkinson's disease. One of those, probably one of the most common, is the Alpha-Synuclein. Is the most well-known. Families that have Parkinson's disease, I mean, that is transmitted to generations, were investigated to find where there is a gene that is triggering the disease. And a long time ago, the problem was identified. There was a mutation in chromosome 4, and after sequencing and investigating the pedigrees in the family and so on, it was the alpha-synuclein gene was identified. And what happened is that there is a difference in one amino acid between healthy and Parkinson's disease individuals. There is only one change right here, from alanine to threonine. There is one nucleotide change, and that change in that letter, in that nucleotide, is enough to trigger the disease. Okay, so that's now we know what was the reason behind this, and we can, once we know the mutation, then you can make mouse models or fly models, or you replicate the disease, and now you have a model that you can use to understand the mechanisms underlying the disease, or to try to discover drugs that can be useful to cure, right, your fly model or your mouse model or whatever you want. So using the same rationale as for the Alzheimer, let's say, okay, this is the alpha-synuclein, which again, there is no alpha-synuclein in flies, which was good. So we can basically bring the mutant alpha-synuclein from humans, put it into the fly to see what happened, and this is what we get. Okay, so you have locomotion problems. You know, you have flies cannot. They are not stable. They're just trembling all the time. You know, they can roll over and see, as you can see there, and Now these flies cannot move properly. They are in constant shaking all the time. They don't fly, actually. We're taking the video from the top, and we don't have any surface that prevents, that they escape or they go away, because they cannot do that. If you try to do this with a normal fly, they will fly away immediately. Okay? So this is just an example of how we can make models of human diseases in the fly. So what's the future of this? What do we want to do with these models? Well, we have models for Alzheimer, Huntington, Parkinson, ALS, and so on. So there are other researchers that are trying to generate thousands of compounds, that are not known. They are mixing using medical chemistry, or actually some teams are trying to get compounds from the ocean, from many parts, or from plants, and then we have the ability to take these thousands of compounds and give it to the fly, So imagine that we have, as I said, we can have literally thousands of flies in different vials. We can use one vial, give them food, and give, let's say, compound number 1, compound number 2, number 3, number 4, and so on. And we just feed the flies, the sick flies. So we know the sick flies cannot move. We can just let them there, let them there, feed them for, 2 weeks and see what happens. And let's see after 2 weeks whether now there is a vial where we can find flies that are moving perfectly. If we find something like that, then now we can go to preclinical trials, or understand what's the nature of that compound and what is the chemistry of that compound, and something like that. So this is where we want to go. Okay, so this is basically an overview of what we are doing in the lab. So in conclusion, the fly is a very flexible organism. We can understand we can study many fundamental problems in biomedical research. Many aspects of human biology are also present in flies. And it's a valuable organism to discover drugs that could eventually prevent or block diseases. Okay? So, any questions about this? So, if not, we can talk more about it. So let me just give you in the next 5-10 minutes an overview of my journey. Okay, then we can talk more about this or the next part. How did I get here? Okay, so I grew up in this small town known as Tampico in the East Coast, in the Gulf of Mexico. So I went, as Dr. Beatriz said, I went to Reynosa. It's a small town in the border with Texas to get my bachelor degree. I'm a clinical chemist by training. So after getting my bachelor degree, I spent several years working in a hospital, in the clinical lab. Okay, so my tasks were getting blood, urine, feces, and investigate, the bacteria or the red blood number or all these things. So I was doing that for several years, and every single day was the same. So I started, at some point, to question, I kind of don't want to do this for the rest of my life. But I didn't have options. Okay? I didn't know research was not an option for me. I never thought, okay, I like science. But I knew that when I was in a clinical lab, I started to associate some findings, like, okay, so there is particular bacteria here, and these are coming from people that are living in that particular neighborhood. And these are all kids. I started to connect things of what I was reporting. And I remember I used to talk to my supervisor. Hey, I think there is something going on here. I think maybe the water in that area is bad, or we should tell someone about this. And then he was, Diego, don't say anything. That's not your job. You don't get paid for doing that, so you just do what you have to do, report it, and that's it. So I felt frustration because I knew things were going on and I couldn't do anything about it. So I didn't have libraries, the internet was not available of course, all this. So I wanted to learn more, okay? So that's how years later I went to a big city, one of the major cities in Mexico, Monterrey. And I enrolled into a master's degree program in microbiology because that's what I was doing at that time, the bacteria and microbia and parasites and all this. So shortly after I got into the master's degree, I was exposed to a seminar given by a new faculty that was coming back from France, and that has had a tremendous impact in my life, and I told you in a little bit. But after got my master's degree and PhD, and I went to Baylor, and then from UTMB Galveston, and finally in UF. But it was in a setting like this, in a seminar, that when I was studying my master's degree, I was exposed to a seminar given by Hugo Barrera. He was one of the first molecular biologists in Mexico. I'm talking about I think he came back in the late 80s. And I went to this talk. We had a class on microbial genetics, and then he came to talk about DNA and all that. So it was my first semester in the graduate school. And when he started talking about isolating the DNA from cells and the facility to running on the gel and visualize it and cut genes and manipulating, and that, at that time, I was like, what is that? What is that? I mean, never heard or never saw anything about that. So that, I mean, it has a strong impact in my life, so that I went, and he went to get that talk because he was recruiting students. So I went, Dr. Barrera, I like a lot, and I like to be part of your group and so on, and that's how it started, okay? But that was the best decision in my life, and he was very supportive. I have a good fortune to have him there, and it was still a role model for me, okay? So when I was working with him, I got the opportunity to develop many skills, of course, many laboratory techniques, self-reliance and independence, and how to think analytically, how to criticize your experiments, what you're doing, how to become more creative, when you're working in science, how to solve problems, organizational skills, multitasking, how you can do different projects at the same time, how to work with others and communicate research with peers and other people, and networking, and there are many other things that were offered to me. This is the kind of environment or skills that I make sure my students get also. So that's I have a personal interest in making sure that whoever comes to my lab, they leave well prepared, okay? Because I got that information that I had an obligation to pay back somehow, okay? So then after my graduate tenure, then I went to Baylor and spent a few more years there working on, now as I told you, on developmental biology, and then I started to work in neurodegenerative diseases. We started to make the first models of these diseases, and actually by the end of my postdoc, my boss was submitting an international patent for the first fly model of Alzheimer's disease. So that was a nice thing that we did. And then, so I was sure that I was ready to begin my own group and to have my own lab, and that's how I got a job at the University of Texas Medical Branch. It was pretty nice. I mean, I have my lab somewhere there. Actually, we could literally go to the beach and come back in 2 hours, or people go and run and so on. So everything was nice. You know, I was so happy. I have a group of people. We have grants and money, but then a few years later, something unexpected happened. Okay? So there was this Category 4 hurricane coming directly to Galveston, and that was a devastation. Okay, so many buildings were totally affected, the flooding everywhere. We lost. There were fires, and there's just, you know, students getting things from campus and the lab. So everybody has. Well, a lot of people had to leave, including myself. Okay? There was no way that we could do research. We got. It was an emergency, general emergency. We couldn't. I lost animal models. I got transgenic animals. I lost samples, antibodies, many things. Okay, basically years of research and effort were gone. Okay, so that was a very devastating situation. So I had to start all over again, and that's how I ended up right here at the University of Florida in 2010. So how can I tell you about, I mean, if someone asks me, will you do it again? I say yes, you know, because I like a lot what I do. I cannot see myself doing something else, you know. So this is my hobby. I get paid to do what I like, and I enjoy it a lot. So there are many advantages of being a scientist. One of these, for instance, I can say, well, you travel a lot. Okay, you travel all over the world, and as a student, usually you're a graduate student, your mentors send you to trips, and they pay for your trip, and so that's nice. There are many advantages, but one of important to me is that every day is different. So it's no routine in this field. I mean, every day is different. Every day you deal with different problems or different situations. So it's always, it was something that I needed. You know, in my case, it's a nice advantage. Of course, there are some, I call disadvantages to me, make me stronger when I have these things. But for other people, maybe disadvantages. There's the stress, okay? Because you never know if at the other side of the world there is somebody working exactly in the same thing you are working on, right? And there is competition always competition to be the first, always. Okay, who's the one who created this or published this or that, you know? And that is what leads to stress. And you have to work hard. You want to be the first to do something, to achieve something, then you have to work hard. And I remember at that time, you know, when you want to do something really hard, you want to work hard, but then you have to do some kind of sacrifices. Sometimes family, you know, so you have to make arrangements with your family, or say, okay, I have to go back to the lab, I have to work tomorrow, it's Saturday or Sunday, you know, things like that. But it's easy to deal. Once you communicate these things, it's not a big deal. And some people claim that there are some limitations, that because you have to work so hard, then you cannot be with your friends, and there's more social isolation. I don't think that's an issue. I mean, you can always arrange and balance your life, and that's why it's important to have good organization and multitasking skills. Because if you plan your day or your week, you can enjoy everything and balance life and work, you know. But so don't be intimidated about being a scientist. I think is great. You need passion. That's the number one thing. When undergrads come to my lab for an interview, and they say, I don't have any experience, I've never been in the lab, I don't care. I don't care, to be honest. I don't care at all. As long as they have passion for what they do and what they like, that's the number one thing for me. So don't worry about, oh, I don't have any experience, or I don't want to make it. You have it here, you'll get it. Okay? So just try to find the right mentor, try to find the right environment, and just work hard, and you will get it. You know? So remember, passion is number one. So finally, if someone asks me, what would you do different? I would say start earlier. Okay, because I waited too much, many years, so there's no need to do that. So you are in the right time, at the right moment to begin. If you think you have something that makes you feel, a scientist, or you want to become a physician or a doctor, that you want to go that way, that's fine. But there are many doctors that also do research. Okay, you can do an MD, a PhD, double degree if you want. So don't turn away from science if you think that you have something there, because it's a wonderful and amazing career. So I think I will stop right there. So you have questions, we can discuss, or comments. Thank you.